We are going to discuss by the end of this lecture, you will be able to know the modes of bacterial genetics and types of bacterial mechanisms for transfer of genetic material, which is DNA, within the bacteria and also between bacterial cells. So the key terms that will be coming across will be mutations, transformation, transduction, conjugation, and plasmid and transposons. So what is genetics? It is actually the study of hereditary characters, but how differences between individuals are transmitted from one generation to the next, and how information in the genes is used in the development of the functioning of the adult organism. But here we are going to talk about bacterial genetics, how these bacteria are acting, because bacteria are haploid. They have a single chromosome and they have a single copy of each gene, which is different from eukaryotic cells, which are diploid and having a pair of chromosomes, having two copies of each gene. One gene is dominant and the other is recessive. So obviously, if the eukaryote cells are different from prokaryotes, so their mode of transfer as well differs. This is the cycle of bacterial chromosome. In replication is a pictorial representation. First, the general features of gene transfer in bacteria. First, it is a unidirectional transfer from donor to recipient bacteria. And the donor bacteria does not give the entire chromosome, but only part of genes. And this gene transfer can also occur between species, which leads to resistance. And this genetic transfer results in genetic variation, and it is also needed for evolution. So, in genetics, we'll talk about mutations, which is mutation is the transfer of DNA within bacterial cells, and this transfer of DNA among bacteria themselves. So, what is a mutation? A mutation is a change in the base sequence of DNA that can result in the insertion of a different amino acid or stop codon into a protein and appearance of an altered phenotype. And mutations results from three types of molecular changes. In the recent edition of Levinson, you will see they are mentioning two types, but transformation is not included in this. But broadly speaking, there are three types, which are base substitution, frame shift mutation, and transposons, which are also called insertion sequences. So first come first, the base substitution. In this mechanism, the one base is inserted in place of another, and it takes place at the time of DNA replication because either the DNA polymerase makes an error or because a mutagen alters the hydrogen bonding of the base. Being used one base inserted in place of another. It takes place at the time of DNA replication because DNA polymerase makes an error. So for the base substitution, the two types of missense and nonsense. Remember, missense there will be a different amino acid insertion, and nonsense will be premature completion of the protein synthesis. Okay, the mutations are change in the genetic material, and the factors involved for that can be chemicals, radiations, and certain viruses as well. Chemicals like nitrous oxide gas and alkylating agents, they alter the existing bases so it forms hydrogen bond. That's why they are called hazardous material. Even if you go near if you're working with these chemicals, they're always hazardous and have got a carcinogenic potential. And they can lead to preferentially wrong base insertion, like adenine. Instead, adenine will be pairing with thymine normally, but after this mutation, adenine will end up pairing with cytosine. These mutations are also by chemicals like 5-bromouracil, which resembles the normal basis and having a less hydrogen affinity, hydrogen bonding fidelity that does thymine. And because of this bromouracil, the hydrogen bonding changes and it binds to guanine with greater affinity. So there is a change in the normal mechanism. So adenine will not be binding with thymine and guanine will not be binding with cytosine. And examples of such mutagens are in our normal life are tobacco smoke, in which there is benzpyrene, which is hazardous for health, and It causes frame shift mutation. Then there are other carcinogens, and they are also having potential of mutant production. For example, X-rays and UV rays. These are other factors causing mutations. Now, what's conditional lethal mutations? Conditional lethal mutations is of medical interest because they may be useful in the vaccine production. As you can see, even in the recent pandemic, how quickly this coronavirus vaccine was made, and because the molecular mechanisms is being exploited for production. And conditional increase indicates that the mutation is expressed only under certain conditions. Most important conditional lethal mutations are temperature sensitive ones, and they can relatively replicate in a low permissible environment like 32 degrees centigrade and function. And one such example is also there in influenza virus vaccine, which comes every year. And this vaccine contains a virus that cannot grow at 37 degrees centigrade and hence cannot infect the lungs and cause pneumonia. This capability of this virus is been done by mutation. But this same thing can grow at 32 degrees centigrade and can reside and replicate in the nose to induce immunity. So it will initiate um, molecular activation or immune activation without causing infection. Now, how transfer of DNA occurs within the bacterial cells? There are three main processes of genetic recombination by which prokaryote fragments of homologous DNA from a donor chromosome are transferred to a recipient cell. They are mainly transformation, conjugation, and transduction. In the recent Levinson book, transformation is not included and they mainly talk about conjugation and transduction. So we'll individually get into the details. This is a pictorial representation of transformation, transduction, and conjugation. We'll come to it again. So first is conjugation. Conjugation is a process of genetic transfer that involves cell-to-cell contact. And direct contact between these two conjugating bacteria is made by a pilus, a fertility pilus or a sex pilus, and the cells are then drawn together for the actual transfer of DNA. As you can see here, this bacteria with fimbriae has produced a pilus towards a recipient bacteria to transfer the genetic material. And it involves a donor cell which contains the conjugative plasmid and a recipient cell which does not have it. And many genes are involved in this synthesis of surface structure, which is the pilus. And only donor cells have these pili. They make specific contact with the receptor on the recipient cell, and then they retract, pulling the two cells together. And these contact between the bacterial cells become stabilized and probably from the fusion of the outer membranes. And this DNA is then transferred from one cell to another. This is conjugation. So, DNA is directly transferable and plasmids carry genetic information necessary for conjugation to occur. And only cells that contain plasmids can act as a donor. Require direct contact between the donor and the recipient bacterial cell. Again, it's a electron micrograph showing fertility plasmid adjoining another recipient bacterial cell doing conjugation. Now, the second mechanism we are going to talk about is transformation, in which genetic recombination occurs and a DNA fragment from a degraded dead bacterium enters a competent recipient bacteria, and it is exchanged for a piece of recipient's DNA. It involves 4 steps in which at first the donor bacterium dies and is degraded, and a fragment of DNA from the dead donor bacterium binds to DNA binding protein on the cell wall of a competent living recipient bacterium. And as you can see here, this is the dying bacterium, and the fragment of the DNA are getting out, and they are being taken up by an actively dividing living recipient cell. And this fragment of DNA from the dead donor bacterium binds to the DNA of a living dividing cell. Then it gets incorporated in the bacterial chromosome, and these are usually done by the presence of RECA proteins, which promotes genetic exchange between a fragment of DNA and recipient DNA. There's another view of the same process transformation where you can see the fragments of the donor DNA coming here, and on the other side, this is the bacteria, the recipient one, which is going to take up these fragments and incorporate it into its chromosome. Now, the third one, mechanism of bacterial exchange of genetic material is transduction. And transduction is a transfer of gene from donor to recipient by the help of a bacteriophage. And what is a bacteriophage? Bacteriophage is a virus that infects the bacterium. And so in this process, the DNA fragment is transferred from one bacterium to another by a bacteriophage. As you can see here, bacteriophage is a virus with a head and tail and contractile sheath, a typical morphology of a virus. And bacteriophage are obligate intracellular parasites, and they multiply inside the bacteria by making use of small or all of the host biosynthetic machinery. And these viruses are the ones infecting bacteria. Again, there's a picture of bacteriophages with having a head, neck, and there's a protein core and a nucleic acid core. And these phage particles, they may have their own nucleic acid and segment of the host DNA. And when the phage particle infects another bacteria, DNA transfer is effected and the recipient cell acquires new characters coded by the donor DNA. Again, this is showing the process of transduction where the phage got attached to the donor bacterium, got inside the bacteria, utilized its replicative mechanism for its own good, and produce many copies of bacteriophages which are carrying the donor genes, and they get out of the bacterial cell and cause mutation, rather transfer of genes. There are two types of bacteriophages. They can be lytic or virulent phages or lysogenic or temperate phage. A lytic phage is the one that multiplies within the host cells, then lyses the cells and release progeny phage. On the other hand, the lysogenic temperate phage, it can be either via the lytic cycle or it enters a quiescent state, which is a resting stage in the bacterial cell. And this is the mechanism responsible for changing certain non-pathogenic organisms into the pathogenic one. For example, diphtheria toxin and botulinum toxin and cholera, these are diseases in which only the toxigenic potential containing bacteria are the ones which are pathogenic, and non-toxigenic strains are considered just the colonizers. They don't cause the disease. Okay, then broadly speaking, types of transductions can be divided into two types. If there is potentially any donor bacterial gene is transferred, it's called a generalized transduction. And if it is occurring only certain specific donor genes, then it's called specialized transduction. And in generalized transduction, the bacteria is infected with a phage, and during a lytic infection, enzymes which are responsible for packaging viral DNA into the bacteriophage. And on lysis of the cell, these transducing particles are released along with the normal virions. The lysate contains a mixture of both, of normal virions as well as debris, which cannot lead to normal viral infection. So here you can see this is infection of donor, there will be phage replication, degradation of the host DNA, the release of phages, and infection of the recipient, leading to legitimate recombination. Now coming to the term plasmid. Plasmid are genetic elements that replicate independently of the host chromosome. They don't utilize they are not dependent on the host chromosome, they are independent. And unlike viruses, plasmids do not have an extracellular form, and they can exist only inside the bacteria and simply as nucleic acids. And that's why viruses and plasmids are sometimes hard to differentiate. So plasmids are fragments of DNA in the cytoplasm. There are different types, like R factor is the one which confers drug resistance. And the plasmids also have bacteriocins, which are proteins toxic to other bacteria and even human cells. And many plasmids are found in gram-negative bacteria. This is a picture of plasmid. We already talked about this is another picture showing the same process, in which there is autonomous replication and there is gene transfer from one cell to another. These plasmids can act as vectors in genetic engineering, like they are also present in certain yeast and gram-negative bacteria. So the functions of plasmids are antibiotic resistance, bacteriocins production, enterotoxin production in certain gram-positive bacteria. Then they also have enhanced pathogenicity and they have reduced sensitivity to mutagens. They are also capable of degrading complex organic molecules. Mutation and recombination, we previously discussed about types of mutation, which and mutation is an inherited change in the base sequence of the nucleic acid comprising the genome of an organism. And these mutations bring about only a very small amount of genetic change in a cell. And mutant is the organism whose genome carries a mutation. Depending on the mutation, a mutant can be maybe showing it or having an altered phenotype from its parent cell. Lastly, we have programmed rearrangements in the bacteria, which are movement of genes from inactive storage site into an active sites where they can express as new proteins. Medically, it's very significant because bacteria acquire new proteins or new antigens on their surface and they can easily evade the immune system. And example of such mechanisms in the bacteria are, for example, Neisseria gonorrhoeae, which causes gonorrhea, and Trypanosoma brucei, and a protozoan that causes African sleeping sickness. These pathogens exploit this mechanism of programmed rearrangement. Now, coming in details of mutations, which is a random, undirected heritable variation. It is caused by alteration in the nucleotide sequence at some point of DNA, which can be due to addition, deletion, or substitution of one or more bases. Mutations can also be divided according on the basis of being selectable, non-selectable, they can be spontaneous, or they can be induced by certain agents. As you already know that bacteria multiply by asexual binary fission. The altered nucleotide sequence expresses new or altered characteristics. It has selective value to the organism and also has got evolutionary value. It acquires antibiotic resistance, grows in body without inhibition. There is also expression of phenotypic variation occurring when genes changes in response to environment, but they can be reversible. Example for that will be certain streptococci have got capsule formation in enhanced form, but the non-capsulated strains are there also, which are not pathogenic. Now again, we are discussing the mutagenic agents, which are some radiations like ultraviolet rays and X-rays, then certain chemicals, alkylating agents, nitrous oxide, benzpyrene, and bromouracil. And certain viruses are also having capability of causing mutation. So in summary, these are the things we discussed, that what are mutations, and genetic transfer can also occur within bacteria by three mechanisms: transformation, transduction, and conjugation. So in conclusion, when bacteria divide, they form clones of identical cells. A bacterium can transfer its genes to another bacterium by transformation, transduction, or conjugation. Plasmids are small bacterial DNA that are independent of the main chromosome. They are autonomous. And transposons are movable stretches of DNA that can jump from one place to another. Thank you.